Hi, everyone. Thanks again for tuning into Benchling's first webinar for iGEM 2020. We're really excited that you guys could be here, and we're even more excited that the iGEM competition is still going on this year. We're really excited to see all these fun projects you guys come up with, and we're here today to introduce you to a platform that all of your iGEM teams could be using. Now, first, I just want to introduce myself and my co-presenter today. So I'm Johnny Trung. I'm a part of the marketing team here at Benchling. Before Benchling, I recently completed my PhD at UC Berkeley in chemistry and chemical biology, where I was studying and engineering RNA elements called riboswitches. And I'll let my co-host, Madeline, also introduce herself.
Hi, everybody. My name is Madeline. I am a customer success specialist here at Benchling, located in the Boston office. So hello from Boston. Previously, I had a bachelor's degree in integrative neuroscience, a master's in biotechnology, so lots of experience there, and I'm really excited to dive into the platform with you today.
All right, and now for the rest of the talk, we're actually going to turn off our videos to be a little less distracting, especially since Madeline will be giving a live demo, and we'd love for you to give a little more attention to some of the content we have prepared today. Okay. Now, so I just want to give you a brief outline for this webinar. I'm going to give you guys an introduction to Benchling because some of you guys may never heard of it before, and then we'll also explore our partnership that we have with iGEM. And then additionally, I'm going to describe a few key benefits of the Benchling platform, and then I'll turn it over to Madeline to give you a full live demonstration of the Benchling platform and all its amazing capabilities. And then we'll close it off with a dedicated Q&A session. And additionally, I just want to go over some quick housekeeping rules for the Zoom software we're using today to do our webinar. You know, we'd love to make this interactive and fun and get to know you guys a little bit better. So we're going to be using the polling feature here today. We'll have some short and really fun, informative questions that we'd love for you to participate in. Additionally, some of you guys may also see the Q&A button on the bottom of your screen. So some of you guys have been using the chat feature, but if you have any specific questions, please put them in the Q&A button so that we can publish them for everyone else to see. Now, so just to get you guys really used to the quick polling feature, I'm going to start off with a really quick poll for you all now. So we know 2020 is a year that's been full of surprises and first experiences. So for those of you watching today, are you actually new to your iGEM team, and will 2020 be your first iGEM competition? So I'm sharing the poll now. Hopefully you guys can see it on your screen. Give you guys maybe, oh.
Hold on one second, Johnny.
Oh, sorry. Oh yeah, sorry. Is there anything?
Thanks, I just wanted to shut my video off too.
Yeah. Sorry.
No problem.
Okay. All right. Awesome. Okay. All right. So it's been about 30 seconds. I'll just end the poll just now. All right. I'm going to share you guys some results. So yeah, that's amazing. Almost 90% of you guys are all new to the iGEM competition, and that's great to hear. So that means we're really excited for you guys all to start your first iGEM competition, and you guys are eager to learn more about it. And so just a quick reminder is that we are going to be doing this is just the first of a series of webinars that Benchling is putting on. You know, and getting familiar and really understanding the power of Benchling really takes time and practice like anything else. And that's why we've dedicated multiple webinars to help you all along the way and share our best tips and tricks. By the end of the series, you'll feel confident in using Benchling for iGEM or any other research project. Since many of you haven't heard about Benchling before, I'd love to give you some small background about it. Our company's CEO and co-founder, Saji, actually worked in a research lab where he was really frustrated by the lack of modern science tools. He found them outdated, disparate, and that they really hindered his ability to collaborate with others. So he started Benchling to really tackle those problems, and since then it's been our mission to fundamentally change how research is being done. Right now we have over 190 employees, over half of which work directly with Benchling users like yourself and also have backgrounds in science. Additionally, the vast majority of our users are academics. We have over 180,000 academic scientists who use Benchling to support their research. And we also support industry. We work with over 270 customers in biotech and pharma, and to help their products help their products go to market. Now, for the second year in a row, we've actually decided to pursue a partnership with the iGEM Foundation to bring cutting-edge software tools to power synthetic biology research. We do this because iGEM and Benchling share a set of core values. We aim to increase microbiology knowledge, spur innovation, and foster collaboration. Now, I'm sure by now you've seen a lot of this logo a lot, and so actually let's take a quick poll to see what you all think it is. It's one of our most common questions, believe it or not, that we get from our users. So I'd love to see if some of you guys actually, know yourselves. So I'm gonna launch this poll really quickly. All right, so we'll give you like 30 seconds or so, and don't be afraid, there's no bad answer. So just love to see what you guys can come up with. All right, 30 seconds. Okay, so I'll share some of the results of that poll. So it sounds like a whopping, a lot of you think it's a jellyfish. Some of you definitely think it's DNA, and you're not wrong about that part, but definitely for those of you guys who shows jellyfish, you are correct. So but more than just a jellyfish, this jellyfish actually has a name. So I'd like to introduce you all to Benchling's official mascot named Jeffy P. So I think Jeffy P helps new and existing scientists just like you make research discoveries faster. And I'm sure a lot of you thinking, well, why a jellyfish and why is his name Jeffy P? Well, the inspiration for our mascot actually comes from a naturally bioluminescent jellyfish species called Aquaria victoria. So some of you guys may have known this jellyfish before. It's scientists were actually able to isolate GFP, aka green fluorescent protein, from it. It's the first fluorescent protein to become really a hallmark research tool in biology. And in the way GFP has revolutionized the way we do microbiology research, we believe Benchling is doing that as well. Benchling is really built for all kinds of scientists, and that includes iGEM teams. At the heart of our software, we really empower teams through our two main applications, Notebook and Microbiology, and which both enhance collaboration. We have a number of features for each applications by themselves, and they'll actually explain how to use them in later workflows today and in our future webinars. I especially want to point out there are exclusive features for iGEMers that we've built. Like an HTML export option. This is especially important for some of you who are creating your iGEM wikis, which you'll need to submit for the iGEM Jamboree. And then additionally, a premium benefit that we've given to iGEM teams is Benchling Sync. It's a way you can edit and update file attachments on Benchling in real time. And on a high level, I just want to summarize a few key benefits that iGEM teams get when they use each of these applications. For the Benchling Notebook, it's more than just an electronic lab notebook and more than a digital version of paper. We've seen that it helps preserve knowledge within iGEM teams between new and old members. You can search for experiments that were done a year ago or just hours ago. There are also multiple ways you can collaborate with Benchling. You can edit protocols together, paste data from different experiments, and you can even create experiment planning calendars. Also, every entry contains a version history. This allows you and your teammates to track edits by each person. You can easily follow up with someone if you have specific questions about an experiment. Now, microbiology serves to be a leader in sequence design and visualization. We offer a unified interface that compiles over 10 design tools onto a single screen. So you're no longer working with multiple software programs open or dozens of browser tabs. We also accommodate a lot of complex workflows. So you can use Benchling for microcloning to CRISPR and even sequence alignments, just to name a few. We also accommodate many complex, we additionally standardize the way you edit and share sequences across your team. You can eliminate these tedious back and forth emails, and you can spend more time in, designing experiments. All right, so now I'd like to turn this over to Madeline to give you a live demo of the actual Benchling platform.
Thanks, Johnny. I'm excited to be walking you all through a high-level overview of Benchling's Notebook and Molecular Biology Suite. We'll also cover how to create or join an organization and set up project folders and permissions. We do prefer that everyone just listen to this walkthrough, although there will be a recording of this webinar if you'd like to rewatch and explore Benchling more independently after the session ends. Let's dive in. You'll notice that once logged in, you'll be directed to the home page. I have a small panel here showing me some of the achievements I've made in Benchling so far, well on my way to unlocking 1 gig of free space. I have recently opened available if I want to navigate directly to a sequence or maybe an entry that I've recently been working on. I also have a projects tab, which I'll be working mainly in today. Our global search tool is useful for searching entries or sequences of interest across projects. Next up, we have the global create button, where you can quickly create a notebook entry or sequence. We do have the ability to connect to external sources such as Google Drive to make accessing your experimental data not living within Benchling easier. And finally, to join a new organization in Benchling, I need to navigate to JeffEP, which represents your user icon. You'll notice that I have the join organization option, and clicking on this, a new window opens where I can start my search. So if I wanted to search iGEM, you'll see I have a few different teams or organizations already created. And if I don't see my organization listed or know that I haven't, my team hasn't created one yet, I can create one down here. Clicking on this, I just need to give my organization a name. So maybe iGEM 2020 test org. Give my organization a handle. I can associate it with an institute or a university name and add my avatar here. Now you'll see that I have invite your organization members. However, we do encourage you and your teammates to sign up for Benchling on the sponsor page for iGEM to ensure you get all the perks of participating in the competition. Next, I can share my current projects that I'm working on. However, I don't really have any projects that I've created yet. So I'm going to navigate to the projects tab now and create a new project. You'll see I have a few projects in here that have already been created, but to create a project, I'll click this blue plus icon. I'll need to give my new project a name. I can select a new owner if I'd like, but I'm going to leave the owner as JeffEP. I can make a description. And then here's where I can manage collaborators. So as soon as my team members join, I can add them here. I can search by name. So maybe I want Johnny to be a collaborator here. I have a list that appears and I can add them directly. So you'll notice that I also have some permissions down here, which I can assign as an admin. Write access allows my collaborators to create and edit their own entries within this project. Read access allows them to view entries within a project, but not create or edit entries. And admin access, of course, is allowing us to do all of those things plus change permissions for this project. So admin access is what a user will be given if they initially create a project, but permissions can be switched around if desired. You are encouraged to create multiple project folders, especially if you have multiple projects going on. And creating a new project is a great way to manage permissions between teams as well, since you may want certain collaborator eyes on one project, but not necessarily another. I'll click out of this, and I'm interested in working in the Benchling iGEM shared project. So clicking in here, this is where my notebook entries are going to reside under this notebook tab here. If I navigate into a project that I don't want to be in, just so you all are aware, there isn't a back button in Benchling. So I just need to hover over the projects tab and I can easily navigate this way. Clicking back in, you'll also see I have an inventory tab, which is going to house my DNA, amino acid, and oligo sequences. I can create folders within both of these tabs to further organize my experiments and data. To create a notebook entry, I can click on, again, the blue plus button within my project. You'll notice that I have the option to create a folder here, then I have two options to create an entry. I can either create a blank entry or I can create an entry from template. Templates in Benchling are used to further standardize experiment protocols that are used on a regular basis. Today, I'll be showing you a completed notebook template and what it can look like, but for our next webinar, we can go into detail about creating one. For example, if I select entry from template, I've already created an RNA extraction protocol, and you'll see that I can search for this template directly, or I could scroll. And I've standardized the template name, so I'm going to enter in the date, or the year rather, the month, and the day. And I'm also going to add my name, and this allows easier searchability in the future. I have my project folder already set for me, but I can change it if I need to. Selecting create. And you'll see that I'm able to pull a copy of the entire protocol without writing it out a second time, which saves me the time that it would take to type and retype the entire experiment out every single time it's performed. If I want more real estate to see my notebook entry, I can click on the project icon again to minimize the side panel. And you'll notice that my entry looks a lot like a word processing document, and we hope that this similarity makes it intuitive and easy for you all to use for your iGEM projects. You'll have a lot of the same functionality where I can change my headings to different sizes by clicking into the entry and using this header option. And then this will reflect accordingly on our table of contents here, which is interactive and will bring you to any section you make in your notebook entry. For example, I want to go directly to materials. Clicking on that, it will bring me to the materials section. I'll scroll back up for now. And one of the perks of Benchling that makes it really easy to collaborate, especially in the times that we're in now, is at mentioning colleagues. For example, maybe I want to tag Johnny. And you'll see that if I type his name after an @ symbol, I'm able to click and it will bring up his name here. And one thing to call out is that Johnny is going to get an email sent to him every time I mention him within a notebook entry. And he may not necessarily want 50 emails throughout this protocol, so just be sure to use this sparingly. Nonetheless, it's definitely a great tool to collaborate with others, especially when a lot of the work has to be performed virtually during this time. And besides, like, who really wants to send an email when they can just tag someone like they would on social media and write a comment? You'll also notice that when I type Johnny's name, I got the option to @mention some project folders and a notebook entry. So I also have those options to tag notebook entries and connect those that way. Benchling's best practice for using the notebook is to create multiple notebook entries, for example, using one notebook entry per experiment instead of stringing multiple experiments within the same entry. We encourage this to create greater searching power within a project folder. To give you another option to collaborate with your coworkers or your team members, you can create a read-only share link up here on the share icon. I can generate this share link, send it out to my coworkers or team members, and I can also retract that access by turning off link sharing. I can link out articles of interest, like my reference materials here. I have my original source as well as adapted from articles. I can insert attachments such as this protocol workflow diagram and put materials, or in this case, reagents, in the table format. And scrolling down further, one of my favorite features is our table functionality. And I know it sounds crazy, but I feel like our table functions are a hidden gem. You'll see in this table sample information, I'm able to select the vendor for my sample by selecting the dropdown. And I have a few different options here that I can change. And then once I select my vendor, I can select cells or tissue, and you'll notice that clicking between these changes A3, what that looks like. So number of cells for cells, or tissue source for tissue. These features have all been configured within a notebook template and can be configured in any basic notebook table. So you can really create what we call very smart tables that will save some time in the long run and further standardize the way you're reporting your experimental details. You'll see that I have both checklists and extra dates listed within my protocol. These dates determine how this experiment appears in my calendar. And I can navigate to my calendar by clicking on the home icon and selecting the caret at the top of the home panel. I can see entries I've created across different projects or choose to filter by project by selecting this little caret near filter. I can also adjust the view dates in the upper right-hand corner of the screen. You'll see that my experiment shows up on the dates that I placed within the entry, and I'm able to click into those experiments like so, and check off the boxes from the calendar view itself. So say maybe I'm too lazy today to go into the entry itself. I know that I'm going to be performing these steps. I don't necessarily have to navigate to the entry itself. But if I want to, I can click on the entry to see what I've done so far. And one thing I'd like to mention is that webinar 2 is going to go more in depth about how to best leverage the notebook and all of its features. So be sure to sign up for that one. Now let's say I'm ready to perform some in silico work to sequences. I need to navigate first to my project folder, then to my inventory, and you'll see I have some sequences here, but let's start with how to import a sequence. I can click on the create button just like I did under my notebook tab, and I have some different options here. If I select import DNA sequences, I'll get a pop-up window with 4 different options on how to import the sequence. You can convert files where you can directly drag or choose sequence files to import, search external databases, select chromosomal regions, and import raw sequences. My personal favorite is import from external database. I can choose my sequence. Maybe I want this gene, make some more granular selections, and then select import. And all of the annotations that belong to the sequence in the external database are automatically imported into Benchling with a click of a button. I'm going to close this window and actually open a sequence that I already have in my inventory, which is human HBB. I'm going to minimize my projects tab just so I have a little bit more real estate. And then maybe I want a little bit more description about my sequence. Benchling has the ability to pull information. So it knows that I imported this from GeneBank, and it will give me things like the file name and the more information depending on where I'm importing it from. I'll select sequence map again. And in webinar 3, we'll be talking more about how to manipulate sequences, but I'd just like to go over some basic navigation and what we have in the molecular biology toolbar to get you started. And one thing I didn't call out within the notebook is this split workspace button at the lower right-hand corner. This button can be used to give yourself access to 2 tabs at once or just one, as you see here, which takes up the full page. Just another way to change around your workspace depending on your preferences. So I have my sequence map pulled up on the left-hand side and my linear map on the right-hand side. And you'll notice that whatever I do to the sequence map is reflected in the linear map. So this is a great way to kind of manipulate your sequence, but also see at a high level what's actually going on. I'm going to unsplit my workspace and maximize my real estate. And you'll notice with a highlighted portion of my sequence, I can see specific attributes at the bottom, such as length of the highlighted region, GC content, and melting temperature. I can delete this part of the sequence, such as just deleting it. And I can also add in a sequence. So I've typed 5 Gs. I'm going to select enter and you'll see that reflected here. Clicking on an annotation will highlight the part of the sequence. So HBB exon, I can click on that annotation. Similarly, I can double click on this translation and you'll notice that that part of the sequence is also highlighted. I can increase or decrease my nucleotide size by this little slider on the upper left-hand corner. And selecting the gear icon at the top right-hand corner of the sequence is where I can toggle view options such as open reading frames, enzymes, or primers. To see the annotations that may have been imported with this sequence, I can go to this highest icon on my molecular biology suite toolbar. And you'll see I have a few listed here, but I can also create new annotations if I have customized annotations that I'd like to add, as well as creating feature libraries. If I want to digest my sequence or manipulate it using enzymes, I can use my digest tool. There's a lot of information here regarding the enzymes themselves. So if I hover over, you'll see that I have things like inactivation and intubation. I can choose to run the digest with specific enzymes here at the bottom, run digest, where I can see predicted products and a virtual digest where I can view products and what that would look like on a gel. So that's a great tool, especially while you're working at home. Maybe you want to mimic a specific plasmid digest and see what it would look like on a gel. That's a great feature. I can also create primers either manually or by wizard. Selecting the wizard option, I can adjust different parameters manually, or I can just select a task, either qPCR or PCR. And when I generate primers, I receive a list back where I can choose to select specific primers, save the ones that I want to keep as I've done here. And primer pairs will be here as well. I do have a history where Benchling will show me all of the edits that I've made to the sequence. So you'll see that I have the changes very detailed and listed here. I can show my full history. And then if I've made a mistake somewhere along the line, I can always click back and reset to version. And you'll see that Benchling has reverted to a previous version. I can choose to align my sequence to other sequences in my inventory using Benchling's alignment tool. And if I'm performing a CRISPR experiment, I can design both my guide RNAs and my HR templates within the CRISPR tool, again, with a lot of customizability options there. If I'm done making changes to the sequence and I want to make sure that I don't make any further changes, I can lock my sequence here by navigating to this information tab. And I also have other options here to move the sequence to a different folder or rename it. And last, but certainly not least, is our assembly wizard tool located down here. So if I wanted to create a new assembly, you'll notice that I have 3 different options. I can perform a simple digest and ligate, a Gibson assembly, or a Golden Gate assembly. And similarly to my notebook entry, I can create a read-only shareable link, which I have the option to do in the upper right-hand corner of my sequence. And to wrap up our discussion of the molecular biology suite specifically, I want to call attention to the fact that we do have this great toolbar at the right-hand side of the sequence, but we also have the ability to manipulate the sequence in almost any ways, almost all the ways that we've discussed with a simple right click. So you'll notice if I click and drag, I have a particular part of my sequence. When I right click, you'll see I can create annotations from here, create DNA sequences, translations, amino acid sequences, and more. This flexibility ultimately leads to less time spent for the scientists or yourselves in what we strive to achieve, acceleration of research and development. And then to tie our molecular biology tools back to our notebook, once I'm finished with my sequence and I want to mention it before I do my qualification and quantification, I can @mention, and I can create a direct link to my sequence. So this is clickable and it will navigate directly back. So again, a great way to collaborate and share information between teams. And I hope I've been able to show you a great preview of that. And thankfully, science can still be done while working safely from home, and you can have fun while doing it. If you ever need help while using Benchling, or you're interested in more specific details about a certain part of our platform, we do have a help center down here where you can search for a specific article or browse within a certain application section. For example, if I'm interested in annotating my sequence, I'll just search for annotations, and you'll notice I have a bunch of options here. We are also happy to help using the chat feature. You're directly able to, we are here to directly answer questions if you're not able to find specific answers within the Help Center or within the platform itself. And I believe Johnny has another poll for everyone, so I'll pass the mic back to him to wrap things up.
All right, thanks for that great demonstration, Madeline. So I'm just gonna start off with like one of our last polls and just get a sense for how you guys like some of these applications so far and which ones you guys think your iGEM teams would probably be using the most. So I know we definitely do have a few questions left, and then hopefully we'll have some time. We'll definitely have some time for the Q&A to get those answered. But yeah, so you guys.
Awesome. So excited to see that both notebook and molecular biology are winning.
Yes. Yes. Yeah, it's always great to have both rather than just one. So yes. All right. So it's like most of you guys have ended, or most of you guys have already answered. I'm just gonna end the poll now and share some of these cool results with you guys. So awesome. Looks like definitely more than half of you are really excited about using both of these, and hopefully you'll be able to use them even better after attending our future webinars. Great. Really quickly, so just want to summarize really quickly some takeaways from the live demo. Essentially projects and organizations within Benchling are really key for structuring your research projects and also making sure the right people have access to your data and your experiments. Now, and the Benchling Notebook is actually it's like a word processor, but more. You can do so many things with it. You can auto-populate calendars, you can create templates for experiments, and there's even more features that we'll be covering in webinar 2. Now, the molecular biology suite actually has so much functionality, there's never enough time to go over them during these webinars. We couldn't if we tried, and we have. But starting off with a lot of the features you think your team would use first is one of the best ways to save you a lot of time and a lot of headaches later. And additionally, it's really best when these, basically these applications are the most powerful when they're used together. They actually communicate with one another like Madeline has showed you, and they make integrating your experimental notes with sequence information seamless. Now, finally, again, don't be afraid to get help. We have a dedicated help center full of tutorials that Madeline showed you, and even more, you can actually chat with someone from Benchling if you can't find an answer to your questions in our help center. All right, so I just wanna open it up to some Q&A time. I know we definitely have a lot of great questions. So what we'll do now is we'll try to read off a few of these, and we'll either have me or Madeline kind of field these. So I'll start with one. One question is, does CRISPR specifically refer to genome editing? Is there a tool for designing single guide RNAs for CRISPR interference? Okay. Yeah, Madeline, do you want to field it? I can also field it. So.
Yeah, that's a great question, and we will try to focus on the CRISPR tool more in depth in our molecular biology. We kind of glazed over it today just because we don't have enough time to get to it, but you can design specifically single guide RNAs. Hopefully that answers your question, but we do have a whole CRISPR center CRISPR section in our help center. So if you want to look at that in depth, and we're happy to answer additional questions if you have them.
Mm-hmm. And you can also do gene knockouts or knock-ins. So you can, so depending on what your application is, you can also do both of those functions using our CRISPR tool.
Yep, absolutely.
So Madeline, we're also getting a couple of quick questions about the formatting for naming templates. Some people are curious about whether the calendar feature requires on the naming of your template. Do you want to elaborate a little bit further?
Sure. So the reason that I added the date in the name was just to kind of standardize the template, since many people, say across the team, could be using the same template. However, you don't need the date within the notebook entry name to show up on the calendar. There is a dates tool within the notebook, which you'll see more about in webinar 2, where you can add dates within the notebook entry itself. So that's kind of what dictates where it ends up in the calendar view.
Mm-hmm. Agreed. And for those of you, some people, if you have a different naming or a dating convention, feel free to use whatever is best within your team. I think Madeline was showing one she uses the most.
Exactly. Yeah.
Yes. And then a couple of you had a couple questions if the recording will be shared online. It definitely will. We'll be giving an email to the iGEM Foundation to share this on their website, as well as giving a recording for everyone who's attended here today. Specifically, there's a question about the chat feature. People are wondering if the chat feature is mostly for people in Pacific time working hours, and are there other employees in European time zones? Madeline, do you want to explain that the most?
Sure. Yeah. So currently we have offices in both San Francisco and Boston. So we do have fairly complete coverage there. We are also open to kind of answering questions as they come in. It's not like the chat feature is you send us a question and it gets lost because we're not in working hours, but we're happy to kind of field those as soon as our reps are ready to go. So unfortunately, slightly limited by time zones at this point, but we're hoping to expand our support in the near future.
Another question asks if you can import sequences from the iGEM parts catalog, and what about FP base? And so for that feature, we actually are integrated into the iGEM parts registry. We will also be going over that in Benchling's microbiology webinar, where we'll show you how you can take a BioBrick or a standard part from the registry and import it into Benchling. So yes. And then another question comes about whether or not Benchling is considering to have downloadable software in the future. So one premium feature that you're getting, Benchling Sync, is actually an additional kind of application like that's on your desktop. But currently right now we are a cloud-based platform, and for a lot of great reasons for being on the cloud, especially sharing and collaboration. So, and then lastly, let's see, we have time for a couple more questions. One asks, do we need to link our already created Benchling account to the iGEM competition or account? For those of you who do not have accounts yet, please go to this iGEM 2020 sponsors page to sign up and get your exclusive iGEM perks. If you have an account that is probably created before you've joined iGEM in some way, please just give us go to the chat feature that Madeline showed you, and then you can actually send a request to get your iGEM perks turned on for you. Okay. I have a few more questions. What criteria do we need to use Benchling's free academic version? Maddie, do you want to take that? I guess maybe they're like hardware specs or software specs, maybe.
They're not that I'm aware of. One thing that I will say is if you're trying to associate with an academic institution, we highly encourage you to use your academic email, but you can also sign up with a Gmail account. Johnny, I don't know if there's any specifics for iGEM, but I know you can use whatever email you want.
Got it.
If you're
Mm. Oh, in terms of browsers, do we have any specified or most preferable ones?
Yes, so Google Chrome is our highest preferred just because we update based on their recent releases. However, we do still work with Firefox as well. The only thing, the only browser that we do not work with is Internet Explorer.
Got it. Awesome. Yeah, so all right, guys, I think that's actually all the time that we'll probably have for this. I'm sorry we couldn't get through everyone's questions, but if you still have your question, please feel free to use that chat feature and then send the questions our way through there. There are also some questions that we'll probably be answering in our later webinars, especially for those of you who are asking more about Benchling's notebook features and also our export functions, which we'll go over more in detail. And so actually, the great time to plug, really, that please make sure to catch us at our next webinar where we'll be talking more about that. And then additionally, if you haven't yet, you can feel free to register using this link below. And then also don't forget to follow us on Benchling on our social media. It's a great place to get reminders, news, and even to give us feedback. So yeah, thanks again for attending today. And then yeah, me and Madeline, we'll see you later. Thank you.
Thanks, guys.